Proteins are large biomolecules or macromolecules consisting of one or more long chains of amino acid residues. Proteins function in our body are catalyzing metabolic reactions, DNA replication, responding to stimuli, providing structure to cells and organisms, and transporting molecules. Proteins differ from one another primarily based on their sequence of amino acid, which is dictated by the nucleotide sequence of their genes. The result in protein folding into specific three-dimensional structure that determines its activity. So there are four protein structures. The first one is primary protein structure. It's a sequence of a chain of amino acid. So a chain of amino acid will join with another chain of amino acid and make hydrogen bonding and become secondary protein structure. Hydrogen bonding of the peptide backbone causes the amino acid to fold into a repeating pattern. It will become alpha helix or beta plated sheet. Hydrogen bonding can occur between two chains of amino acid because every partially positive amino group sticks to the partially negative oxygen in the carboxyl group of the amino acid for residues earlier on the chain. So the third one is tertiary protein structure. It is three-dimensional folding pattern of a protein due to side chain interaction. And the fourth one is quaternary protein structure. It is a protein that consisting of more than one amino acid chain. Proteins in our body. First, enzyme support the reactions that occur in the body, act as biocatalysts. Second, structural protein, for example, collagen and keratin. Keratin type protein is a strong, fibrous protein, so can form the structure of the skin, nails, hair, and teeth. Collagen forming tendons, bones, muscle, cartilage, and skin. Third, antibody protein form antibody components in the body. Fourth, transport protein, for example, blood, hemoglobin, and albumin serum. Hemoglobin function is to bind oxygen and deliver it to the tissues that need oxygen from the lungs. Albumin serum, delivering fat to the bloodstream. Fifth, binding protein, for example, ferritin. Ferritin is a protein in charge of iron binding. When later iron is needed again to form red blood cells, the iron in ferritin will be released. Sixth, actin and myosin. Actin is a protein that forms fine filaments. Myosin is a motor proteins best known for their roles in muscle construction.
Classifications. There are thousands of amino acids in the world, but there are only 20 kinds of amino acids that make up the proteins in human bodies. Based on their needs in the human bodies, they are classified into essential amino acids and also non-essential amino acids. There are 9 essential amino acids and there are 11 amino acids. Essential amino acids cannot be synthesized in the body, so we need it through our diet. While non-essential amino acids are synthesized in our body or obtained from protein breakdowns. Essential amino acids include leucine, isoleucine, lysine, threonine, methionine, phenylalanine, valine, tryptophan, histidine, while non-essential amino acids include alanine, arginine, asparagine, cysteine, glutamate, glutamine, glycine, proline, serine, tyrosine, aspartate. There are also conditional amino acids that are needed in times of sickness or stress. They include arginine, cysteine, glutamine, glycine, proline, serine, tyrosine, and one other type of amino acid called ornithine.
So, almost all amino acids are chiral. The only amino acids that are chiral is glycine. The reason why glycine is not a chiral amino acid is because it doesn't have asymmetric carbon atom, which makes it as chiral. And there is also a functional of amino acid. The function of amino acid is to be hormones productions, muscle structure, nervous systems, vital organs working, and cellular functioning.
Protein synthesis. So now, I'm just going to explain briefly about the synthesis of protein. So basically, protein synthesis consists of two steps: transcription and translation. Transcription and translation takes information in the DNA and use it to produce protein. So the first step is transcription, which occurs in the nucleus. DNA, with the help of RNA polymerase enzyme, is separated into two strands, and then a strand of mRNA is built by adding complementary nucleotides to template DNA. In transcription, mRNA is produced. mRNA is the link between DNA and the production of protein. In the translation process, mRNA from the transcription process delivers information from the DNA to ribosomes, which consist of rRNA and protein. Ribosomes will then read the sequence of codons in mRNA, and then the molecules of tRNA will bring amino acids to the ribosome in the correct sequence. The start codon is called AUG, which has anticodon UAC, which brings methionine. The amino acid building process will stop when it reaches the codon UAG, UAA, or UGA. Amino acid will continue to be produced, and then it will form polypeptide chain, and that chain is going to bend and fold on itself to form protein. Denaturation of protein. What does denaturation of protein means? Denaturation involves modifying the molecular structure of protein without breaking the peptide bonds, which causes loss of biological activity or catalytic activity in enzyme, loss of solubility, and communal aggregations. Denaturation only happens in secondary, tertiary, and quaternary structures only, because denaturation aren't strong enough to break the peptide bonds in the primary structure. In denaturation, the protein loses its form. How so? Factors of denaturation causes the weak linkage or bonds, such as hydrogen bonds within the protein molecule, that are responsible for the highly ordered structure of protein broken, so it can maintain its structure. The cause of denaturation includes heating or high temperature treatment, drastic change of pH, such as treatment with alkali, acid, urea or detergent, or vigorous shaking. In case of heating, heat increases kinetic energy and causes the molecules to vibrate so rapidly and violently that disrupts the hydrogen and nonpolar hydrophobic bonds. Examples of denaturation are boiled eggs, cooked meat so it's easier to digest, production of yogurt by lactic acid bacteria, denaturation of hemoglobin. Denatured protein can be turned into its native state again. This process is called renaturation by the removal of denaturing agent and restoration of conditions or environment favoring the native state.
Plasma membrane is a biological membrane that defines the borders of the cell and allows the cell to interact with its environment. There are two membrane proteins, integral and peripheral protein. Integral protein integrated to the membrane may or may not extend through both layers as an ion channel and also carrier or transporter. Peripheral protein is attached on the inner or outer surface as a receptor and also surface antigen or identity marker. There is also a glycoprotein, protein with a sugar attached to it. It involves in cell-to-cell recognition. If there are more amino acids than the body needs or if cells are starving, some amino acids will get broken down for energy via cellular respiration. In order to enter the cellular respiration, amino acids must first have their amino group removed. This step makes ammonia as a waste product, and in humans and other mammals, the ammonia is converted to urea and removed from the body in urine. Once they have been deaminated, different amino acids enter the cellular respiration pathways at different stages. The chemical properties of each amino acid determine what intermediate it can be most easily converted into. For example, the amino acid glutamate, which has a carboxylic acid side chain, gets converted into the citric acid cycle intermediate alpha-ketoglutarate. This point of entry for glutamate makes sense because both molecules have a similar structure with two carboxyl groups.